And so we're really very pleased today to be hosting Dr. Jason Rosenbaum here to present Grand Rounds at Duke and 4 virtual pathology Grand Rounds. Dr. Rosenbaum is an assistant professor at the University of Pennsylvania, Perelman School of Medicine in the Department of Pathology and Laboratory Medicine. He is the director of the Molecular Genetic Pathology Fellowship Program and the director of Solid Tumor Genomic Diagnostics. Dr. Rosenbaum completed his Molecular Genetic Pathology Fellowship at the Washington University School of Medicine in St. Louis and his residency at the University of Wisconsin Hospital and Clinics in Madison, Wisconsin. He is board certified in molecular genetic pathology and anatomic and clinical pathology. Dr. Rosenbaum has worked to develop the novel immunohistochemical and molecular marker, INSM1, for neuroendocrine carcinoma, and improved algorithms for tumor mutational burden and copy number variant calling from targeted oncology panels. He has developed and administers the Penn Cancer Genomics Case Conference and serves on the Association for Molecular Pathology Professional Relations Committees and the College of American Pathologists Personalized Health Committee. Jason and his wife Debbie are the proud parents of 2 boys, Max and Bowen. Outside of work, Jason enjoys spending time with his family, writing, occasional karaoke, and various forms of comedy. He grew up in the San Francisco Bay Area where he wanted to be a cowboy. And so now he's in the wild west of medical genetics. Dr. Rosenbaum, you can share the screen.
Thank you, Sarah, or Dr. Jang. Sorry about that. Thank you to the Duke Department of Pathology and to ASCP for sponsoring this. I'm very pleased to be here. Just a little bit more about where I'm coming from and what you can expect from this talk. I am a little bit unusual in the field in that I'm almost exclusively focused on next generation sequencing. I don't have other clinical obligations. I'm not also a hematopathologist, for example. And I don't have a research component. And so unlike many people that are operating in the field, I'm exclusively focused on next generation sequencing, which gives me a little bit of a different perspective of the global issues. And so even though some of the things I'm going to be presenting may strike you at first as a little basic, I'm hoping that the perspective I offer gives you some insight into a new way of thinking about next generation sequencing. Okay, that's not working. Okay. All right, so we're going to start really basic. What is NGS? Literally, it stands for next generation sequencing, which is not that helpful. The words next and generation don't really mean that much. I'm not persnickety about terminology in the sense that I would never correct you for using the term NGS, and I use the term NGS myself when I'm speaking to people about the technology. But when I'm trying to teach, when I'm trying to convey what the technology really is and what it means, I try to use the term massively parallel sequencing, because the words massively and parallel actually carry meaning, and I think they reinforce what we're talking about. So starting there, where does massively parallel sequencing come from? Massively parallel sequencing owes a huge debt. In fact, it wouldn't exist at all without the Human Genome Project, which in its simplest description was aimed at sequencing the 3 billion base pairs of the human genome. And that was important for a couple of reasons. One is that without the human genome as a reference, we wouldn't be able to do most of what we do in today's sequencing era. We need to have something to compare the patient to. But in addition, the Human Genome Project, much like the moon landing, introduced technologies that were not necessarily specific for the purpose for which they were designed. Shotgun sequencing, of course, existed before the Human Genome Project, But the Human Genome Project made use of shotgun sequencing at a scale that was previously never attempted. Shotgun sequencing is pretty simple. It sounds maybe a little bit intimidating, but the idea is that you take sequenced fragments of nucleic acid from the genome or portion of the genome that you're interested in. You align those fragments to something. In the case of the Human Genome Project, it was really to each other. You looked at the overlapping regions and tried to piece together the fragments. And you use that alignment to regenerate the full sequence. And that's because there is no technology that can just sequence 3 billion base pairs in order at a pop. And so some kind of fragmentation and subdivision of the labor had to be attempted. This was a massive project, and it existed in the context of sequencing technology that was primarily Sanger sequencing, which is fine. Sanger sequencing is, whoops, got a little ahead of myself there. Sanger sequencing is very sensitive in some ways and can sequence, generally speaking, longer segments of DNA than massively parallel sequencing. So what was the problem? Well, the problem is that Sanger sequencing requires running out the sequence, the DNA fragments on a gel in order to resolve the different nucleotides, as you see here. And those gels take up space, and the Sanger sequencing reaction takes up a certain amount of reagent, and that becomes very limiting when you're trying to do a massive sequencing project. And so out of the Human Genome Project evolved these new technologies in order to miniaturize and parallelize the sequencing process. And so into that technology is where we come in the clinical space. So that brings us to the question of why would you use massively parallel sequencing clinically if other technologies exist and they may be more well established. Why would you consider massively parallel sequencing? Well, to return to shotgun sequencing, if we look at it in the clinical space, it's a little bit different. Rather than aligning the fragments to each other and piecing together an unknown, we have the opportunity to align our sequence fragments to the reference because the reference now exists. And from there, we can regenerate the patient sequence. And this is as if you had a giant pile of Legos. You follow the directions and you build a model. And I don't mean this example to be condescending. What I mean it to do is reduce something that is very complex at its core into just a fairly understandable concept. And one of the reasons that I like this particular example, is because I'm a 44-year-old white male, and so Star Wars holds a special place for me. But beyond that, this model, to me, illustrates that the human genome is incomplete and our understanding of genomics is incomplete, and there are plenty of gaps left to fill. So why use it clinically? There are tissue limitations to PCR-based assays, of which Sanger sequencing is one. Each individual assay requires some nucleic acid. In that context, there is an increasing need to test many loci in any given disease. And so you would have to multiply the amount of nucleic acid you needed by the number of loci if you were using individual standalone assays. And so, for example, if we take the IASCLC CAP-AMP guidelines for testing lung cancer, which were issued in 2018, they require or strongly recommend testing EGFR, ALK, ROS1, KRAS, BRAF, RET, MET, and ERBB2, which is the correct genetic name, HGVS name for HER2. And independently of the fact that that is a lot of different genes to test by standalone assays, it also requires detection of different kinds of mutations. So EGFR would require SNVs and indels. ROS1 and ALK would require detection of structural variants, and for resistance, you would also need single nucleotide variants. And MET and ERBB2 also would benefit from having copy number variants detected. At the same time, so in parallel with this pressure to test more things, there is an increasing need to accommodate smaller and poorer specimens. As radiology gets better and better and biopsy techniques get better and better at hitting small targets, the specimens that we're asked to test become harder and harder, more difficult to extract enough nucleic acid, especially if you're operating in a regime of individualized standalone tests. And so when massively parallel sequencing became available, of course, it started out very expensive. And of course, at first, there weren't enough genes to make it cost effective. There weren't enough clinically actionable genes, I should say. But costs came down over time. And it turns out that in many ways, not in every way, but in many ways, massively parallel sequencing provides superior performance characteristics when compared to standalone assays. Again, that's not always true, but it is often true. Depending on the assay design, a massively parallel study may require only as much as 50 nanograms total of nucleic acid. So I want to take a moment here to talk about a concept that should be familiar to anybody that went to business school. I didn't go to business school, but I think it's a fairly straightforward concept regardless. And that is when you're providing a service or a product, you essentially, if you want to oversimplify things, you have three parameters you can try to maximize. You can try to maximize the speed at which you deliver the service. You can try to maximize the quality of the service, or you can try to maximize the cost effectiveness, in essence, minimizing the cost, keeping it cheap. And the principle they teach in business school is that you get to pick at most two of these parameters to maximize. And that's why the corners are highlighted. And so if you try to emphasize speed and cost effectiveness, you end up with a product that is fast and cheap, but it is not very good. It is of poor quality. And similarly, if you try to emphasize both speed and quality, the product ends up being fairly expensive. And this is usually illustrated in the context of food, which should be familiar to any pathologists in the audience. So fast food is pretty straightforward. It's fast and cheap. And whatever you think about the taste of the food, generally speaking, the quality is lower. In contrast, if you went to a fancy restaurant, the food is delivered to you relatively fast, certainly within minutes, on a scale of minutes from when it was ordered. We're certainly not talking about days. But you retain the quality and at the cost of money. And then finally, you can manage both your costs and your quality and maximize both parameters if you're willing to cook your own food and potentially raise the ingredients yourself, grow the ingredients yourself. So that's fine. Everybody basically understands this in the context of consumer goods and of food. But somehow when we enter medicine, people tend to forget this, and pathology specifically. And that center triangle tends to become very attractive. And you hear people saying, well, I want to maximize speed, cost, and quality. And you can do that. You can put your product, you can put your assay in the middle of the triangle. That is certainly possible. But what ends up happening is that it's something of a trap, because any consumer of your product, whether it's an oncologist or a patient or a clinician of some other type, can look at what you're delivering and say, well, I could have gotten it faster somewhere else, or I could have gotten it better somewhere else, or I could have gotten it cheaper somewhere else. You don't hear that one too often in medicine. Most people outside the lab are not too concerned with cost. But in any case, trying to land in the middle of the triangle ends up being something of a trap. And so when you're thinking about assay design, or for most of you who won't be actually designing assays, if you're thinking about how the assay you're using was designed, it's often important to consider whether the priority for your result should have been speed or should have been quality. For those of you that are thinking about designing assays or bringing assays in-house, I would encourage you to try to emphasize one or the other, but not fall into the trap of trying to do everything on a single platform. So back to the question of why massively parallel sequencing. It has some advantages. Literally, it is massively parallel. It generates a huge amount of data in one shot. You can interrogate millions of loci in the genome, and each locus, even if you're just looking at single nucleotide changes, what traditionally people call mutations, each locus can resolve 3 different changes. So that's 3 times millions right there, and that's not even accounting for insertions, deletions, etc. On that note, it also can provide more kinds of data at once, where most single gene assays are only capable of delivering one kind of mutational data, single nucleotide changes, for example, or amplifications or rearrangements, etc. These assays can deliver multiple kinds of data at once. They are what we call multi-parametric. The disadvantages, though, are real, although there's a caveat, and that's why they're in quotes. These assays are slow and they are expensive, but it really depends on making the appropriate comparison and understanding what your real needs are. And so, for example, if you're concerned about myeloproliferative neoplasms, certainly you want to be able to test JAK2 V617F. And it should go without saying that a massively parallel sequencing solution will be slow and expensive. It will be a slow and expensive strategy if all you want to do is test JAK2. And I would be the first person to say you shouldn't employ a massively parallel sequencing strategy if that's all you're trying to do. But not every patient is served by just testing JAK2. There are patients that need additional genes testing. And again, these are additional kinds of mutations as well. And so it's really important when you're thinking about whether massively parallel sequencing is appropriate to think about what you are trying to get at. As a rule of thumb, massively parallel sequencing becomes cost-effective per unit run when it replaces about 3 standalone assays. Now, it may be clear that some of this, particularly turnaround time comparisons, are going to be dependent on whether you test things one by one and say, if JAK2 is negative, then I go to BCR-ABL, and if BCR-ABL is negative, then I go to calreticulin. Massively parallel sequencing by itself may be faster than that strategy. It probably will be. However, if you would do your standalone testing in parallel, the comparison becomes more complicated. It's also important to consider the global cost effectiveness of the platform. And I mean that in 2 ways. One is that the cost effectiveness of the platform being sufficient to replace 3 standalone assays really depends on the assumption that you already have the sequencer and the expertise in-house. It doesn't account for startup costs. And so that's important to think about. And then the other thing to consider, though, is that once you have the platform and once you have the technology, it may become fairly cost effective to start testing for rare conditions that don't by themselves support an individual test. And so right away, we start to get into the triangle of quality. If you're testing a huge amount of the genome, like whole exome sequencing, or a larger portion of the sequencing than panels, you're able to recoup some of those costs by testing for rare diseases that might otherwise get sent out. But if you are doing whole exome sequencing, that has attendant costs and turnaround time consequences as well. So what does massively parallel sequencing detect? We've sort of skipped over that question a bit or alluded to it in previous slides. In order to talk about that, I'm going to lay a little bit of groundwork. What you're seeing here is an image capture from a piece of software called Integrated Genomics Viewer. And that's a tool that many of us use in the sequencing space in order to visualize our data. And so what you're looking at here, all these gray bars are Legos. They're equivalent to the gray Legos that made up the model in the prior slide. And so those are our individual DNA sequencing reads. At the bottom, you see the reference genome. It looks like a rainbow colored strip at this resolution. It's not actually rainbow, it's four colors, one for each nucleotide, A, C, T, and G. And at the top, you see a chromosomal map that gives us a sense of where we are and what the scale of what we're looking at is. The red hash mark in the chromosome indicates the position we're looking at. The red blowout lines I've added in myself. Those are not natively present in the software. One thing I'd like to point out is that we're not very far into the chromosome. This is chromosome 2. But if you look at the base pair measurements, we're already at almost 30 million base pairs. That's a lot of sequence. The genome is big. At the bottom, you see a map of the gene we're looking at. This gene happens to be ALK, and the thick blue bars represent exons. The thin black lines represent introns, and the arrowheads, it may be hard depending on your screen to see, but the arrowheads are running right to left, which indicates the direction of transcription of the gene, essentially which strand of the DNA the gene lies on. Okay, so in that context, nearly any massively parallel sequencing can detect any assay rather, can detect single nucleotide variants. That's essentially what they were designed to do. And it does that by detecting a mismatch from the reference genome. Whoops, went backwards. Some mismatches are highlighted here. They're represented by hash marks. The color indicates what the variant nucleotide is. And you only see a hash mark when the patient sequence does not match the reference genome. And so highlighted in red is something that is called in a number of reads. So it would be what we would, well, I'll get to that in a minute. It's called in a number of reads and they all line up. And so that's supportive of that being a real call, not an artifact. The little ditzels highlighted in purple may or may not be real. They would require a little bit more investigation. But the fact that we're only seeing them in one-off nucleotides in this view is suggestive of the fact that they might just be sequencing error. Additionally, nearly all of these assays are very good at detecting small insertions and deletions. What you see here is how a deletion is represented in IGV. It's a thin black line interrupting the gray sequencing read. In this case, it's 10 nucleotides removed from DNMT3A in a myeloid neoplasm. Insertions are maybe a little bit counterintuitive the way they're represented. In contrast to a deletion, you don't see the whole insertion. What's introduced instead is a purple hash mark where the insertion lies, and you can click on the hash mark in order to get more information about exactly what the insertion is and how long it is. So nearly all of these assays can detect single nucleotide variants and indels. Almost by definition, that's what they're meant to detect. And that's because they're fairly easily detected by comparing the patient's aligned sequence to the reference genome. You can just compare up and down in IGV, and you can see where potential mutations are lying. Now, with appropriate assay design, you can't necessarily assume that any of this is true, but it is certainly true that with the appropriate assay design, you can also detect structural variants, which would include large insertions or deletions, chromosomal rearrangements like EML4-ALK or BCR-ABL1. Copy number changes can also be detected. And one of the reasons that this requires separate design is that structural variants don't necessarily actually have much that mismatches the genome. So if you think about an ALK EML4 rearrangement, for example, the ALK sequence is ALK sequence and the EML4 sequence is EML4 sequence, and they both will align to the reference genome. It's just that they're in the wrong place. And so it requires specific tools and specific assay design in order to make sure you can detect such things. Similarly, in some ways even harder, are copy number variants, because everything matches the reference. Nothing's even out of place. You just have too much. And the example would be HER2 amplification or any amplification, where you just have too much or too little of the sequence you're looking for, and nothing, strictly speaking, mismatches the reference genome. And so that requires special tools that can statistically assess the quantity of what you've sequenced. You can even get fancier. You can look at expression data. You can look at what I call metamutational data, which would be something like a mutational signature. You can distinguish germline from somatic mutations, et cetera. But all of these things depend very much on assay design. And many of them, not all of them, but many of them come with trade-offs. So if you really wanted to be able to distinguish somatic mutations from germline variants, the only way to reliably do that is to sequence paired specimens. And by necessity, that is going to increase your sequencing costs. Paired, by paired, I mean a specimen with the disease and a specimen known to be free of the disease. So you can pretty simply state what does massively parallel sequencing detect by just saying it can detect anything involving nucleic acids given the right assay design. And that illustrates an important point, that it's a technique, not an assay. And what I mean by that is that saying something like the patients should be sent for NGS is about as meaningful as saying the patient should be sent for radiology or the patient should be sent for microscopy. It's not completely meaningless, but it's not, on the other hand, that helpful without knowing what assay you want to run, what exactly you're looking for, what's the clinical question that's trying to be addressed. Generally speaking, very, very broadly, you can divide the clinical questions into two kinds of groups that really define how the assays are built. And these are panels versus exploratory assays. A panel assay is usually driven by a specific diagnosis or a specific condition, meaning something like cancer, something like cancer or even a subtype of cancer. It's a limited set of genomic targets, which constitutes a gene list usually, and it's the more common approach in adult medicine. Exploratory assays, in contrast, are usually agnostic to diagnosis, and they have very broad genomic coverage, often whole exome or whole genome. And they tend to be more common in pediatric medicine. It's less true than it used to be, but it used to be true that if you said that you were doing next generation sequencing in a pediatric context, you essentially meant whole exome sequencing. Whereas in an adult context, you essentially meant panel sequencing. Again, that's less true than it used to be, but it just illustrates the different practice patterns um, are dependent on context. So on the left are some examples of some panels that you might imagine. Here on the right are some ways that an exploratory assay may be deployed. And again, for something like developmental delay or dysmorphology, the idea here is that a pediatric patient presents with an uncharacterized syndrome that doesn't really fit the textbook definition of anything, but is presumed to be genetic. And you're using the assay to help define the diagnosis. Usually in adult medicine, you've already narrowed down the diagnosis somewhat, and you can narrow the target space of your assay considerably. And there are some assays that don't neatly fit into any category, but that's beyond the scope of this talk. So that's what massively parallel sequencing can detect and what it can be used for, but what can it miss? I think this is an important question. It's one that often people in my field are reluctant to talk about because we're often struggling to get recognition for the importance of the studies. And so there does tend to be an instinct to minimize the gaps. But there are gaps and they're important to be aware of. So the first question you ought to really be asking as someone outside the lab is what is included in the panel and what isn't? If the gene that I'm interested in hunting for a mutation in isn't even on the panel, then the test is going to miss it. So this is a representation of the gene list for our solid tumor sequencing panel here at the Center for Personalized Diagnostics. It's 152 genes. Some panels will only be portions of genes. It's important to clarify that. For us, on our solid panel, we sequence the entire coding sequence of all of these genes, plus 10 base pairs into the adjacent introns. Most panels typically don't include non-coding sequence, for example, promoter variants, unless otherwise stated. And so you shouldn't assume necessarily that something like a TERT promoter would be identified, a TERT promoter mutation would be identified unless it's explicitly said in the test methodology that the TERT promoter is covered. There are such things as hotspot panels. Ours is called the Penn Precision Panel, or PPP. These focus on very common high yield single nucleotide variants and small indels. And the genes that are underlined and in bold in the image of the gene list are the constituents of our PPP panel, of our hotspot panel. And so notice that the coverage of our hotspot panel is much narrower just on a gene list basis, but it's also narrower in that not the entire coding sequence of those genes is covered, only hotspot regions. That's an important thing to understand, is that not every assay is necessarily going to cover what you're looking for. And it sounds obvious, but we do run into this problem occasionally where the test is ordered and it turns out that it didn't even cover what the clinician was interested in. Rewinding a bit, going back to our view of IGV, I want to address this question of small indels that may have piqued your interest before. Why are these assays good at small indels but bad at large indels, or not as good at large indels? If we highlight one of these reads and we say for the sake of argument that it's about 100 base pairs long, they're usually a bit longer than that, but just for simplicity, we're going to call it 100 base pairs. If we introduce a deletion into that read that is close to 100 base pairs in length, it doesn't really leave a lot of sequence for alignment. And so the larger a deletion, and also similarly, the larger an insertion, the harder it is for the software to align the read appropriately to the genome, and you get what's called dropout. If the deletion or insertion affects enough reads locally, you get a lot of dropout. And you can maybe start to surmise that something is going on because your assay is underperforming in that region, but no actual variant is being called. You don't know specifically, for example, whether that's even an insertion or a deletion. And so the fact that we have fragmented the fact that we have fragmented the DNA has consequences. That's also illustrated here. This is taken from a report from a corporate sequencing solution. And I think they've done a nice job here of laying out the idea that these assays are often treated appropriately so, like they're a single thing. You put DNA in one end and out the other end you get a report. But in some ways, it's more appropriate to think about these assays as multiple assays being run in parallel, because the assay may be very good at detecting one kind of variant. For example, single nucleotide variants. This assay is 99.9% sensitive. Not as good at detecting other kinds of variants, 98% sensitive essentially for insertions and deletions. And may vary quite a bit, maybe depending on tumor content for other categories. So copy number changes here drop from 99% sensitive to 92% sensitive, depending on the percentage of tumor nuclei. And then finally, maybe even by specific target, the performance of the assay can vary. So this assay is really good at detecting ALK fusions, but not so good at detecting fusions of other genes. 90% sensitivity is okay. It's not ideal. We get this question a lot. Why does massively parallel sequencing take so long? And it's a fair question. Turnaround times are generally on the scale of weeks, not days, and that's to say nothing of hours. So you might reasonably ask the question, we've got two different panels here illustrated by our solid panel and our PPP. You might reasonably ask the question, do bigger panels take longer? Well, sequencing is batched, and if we sequence an assay that constitutes about 150 genes, on our assay, at the depth of sequencing we try to achieve, we can sequence roughly 20 specimens in a batch. If we kept the sequencing depth the same and reduced the number of genes to 50, we would be able to sequence about 60 specimens. But it doesn't change how fast the sequencer goes. The sequencer goes the same fast, the same speed, no matter how many specimens you're running. And if that's a little bit confusing, let's do this by analogy. Ask yourself the question, can the airplane leave an hour earlier if all the passengers lose weight, or if all the passengers left their luggage at home? And the answer is no. The airline schedule is dictated by their expected ticket volume, not by the weight on the plane. The plane may marginally travel faster if it carries less weight, but it's not going to leave any earlier. So do bigger panels take longer? No, that is not the reason that massively parallel sequencing takes so long. What about assay complexity? Massively parallel sequencing, I've reduced it to a very simple framework here, but it's in truth much more complex. There are different stages to the sequencing process, and each of those stages has sub-stages, and there are even intervening steps that I'm not illustrating here. And each of these steps does take time. So acquiring the specimen can be as short as hours, but it may take weeks if you're requesting material from an outside institution. The library preparation takes hours to days. Sequencing can take hours to days. The bioinformatics pipeline can range quite a bit. And interpretation and reporting, just like interpretation and reporting of histology or radiology, can be done quickly under the right circumstances, but can also be delayed for a variety of reasons. So you might look at this and say to yourself, well, if we got lucky, or if we design things very carefully, we might be able to keep every one of these stages to hours, and we might be able to get a turnaround time of 1 to 2 days. And that is technically possible, but and it is a component of the answer, but it is not the whole story. Again, because sequencing is batched. Batches wait to fill. Or they wait until a pre-scheduled time to leave, like an airplane. And the frequency of the runs is dictated, not detected, by the expected case volume, not by any previous steps in the workflow. So unlucky specimens get added to the run first and end up with a slower turnaround time. And your lucky specimens get added to the run last and have a faster turnaround time. And so the real answer to the question of why massively parallel sequencing takes so long is because it's so expensive. We can't just run these assays, generally speaking, one-off, one at a time as they come in. If we could do things much faster. There are some exciting and interesting new technologies that are being introduced that may help ameliorate this problem, but it continues to be a problem currently. So wait a minute, why offer a hotspot panel if it's not any faster than the regular panel? Well, I'm so glad you asked. The reason is not because it's faster to run the hotspot panel. The reason is that the hotspot panel can tolerate poorer specimens, specimens with less DNA or more highly degraded DNA, DNA of poorer quality, may be able to be run on the hotspot panel where they might not make quality cutoff for the bigger panel. Okay, so back to pre-analytics, which can be a huge time hole in this whole process. Let's ask the question why we evaluate the tissue at all. Why can't we just do specimens straight to sequencing without looking at the tissue? And this is in the context of tumor sequencing, not germline sequencing. First of all, we have quantity and quality cutoffs, but those quantity and quality cutoffs are dependent on measurements of the nucleic acid proper. So they're dependent on the input that is given, so the quality or type of the specimen, FFPE versus fresh, for example, or the number of nuclei. So here are images taken from the exact same neoplasm, one of which has more nuclei, more concentration of DNA than the other. And so would represent a better specimen. But the minimums for the assay also depend on the output. What are you trying to identify? If you're just trying to identify SNVs and indels and you can ignore other types of variants, then you don't need as much DNA, and that's the principle behind the PPP assay. However, if you need your assay to detect copy number change or metamutational data like TMB or MSI, you need better quality DNA. All of that is independent of tumor content. And as a way of illustrating that, I would mention that not all assays are meant for cancer. You can use next generation sequencing for cystic fibrosis, for sickle cell anemia, for anything germline, and there is no tumor content for those diseases. So why are we even assessing tumor content? The reason is that it's generally believed to be important for patient care. We don't want to run assays that are unlikely to yield a meaningful result. basically we're trying to reduce false negatives. This prevents unnecessary delay of treatment, and it allows you to pursue alternative specimens or alternative testing if that's warranted. So what is it that we're assessing? Well, all assays have a lower limit of detection, and that is represented as a tumor percentage. Some quick math: if you're covering 150 genes, that's about 500,000 base pairs. If your assay it has a mean depth of coverage of about 1,000x, that's 500 million base calls per specimen. If we assume the error rate is super good, that we're only wrong 1 in 1,000 times, then that's 500,000 errors per specimen. That's enough that there could be 1 error per every position in the assay. Now, we're not just sequencing the assay, we're not just sequencing the specimen 1 time, we're sequencing it multiple times. So if the errors were evenly distributed across the assay, that would be okay, but they're not. They're concentrated in certain areas. And what you're seeing here is an image that just represents the background sequencing error of the IDH2 R172 locus. And so you can see that if we tried to detect mutations below a certain level, it would just be impossible to distinguish a real mutation from the background sequencing error of the assay. And so that's why we end up with the lower limits of detection, the tumor percentage cutoffs that we do. It is important to point out that it would be unreasonable to formally validate every mutation at every position of even a medium-sized assay like our solid assay. And so validations for these assays are a little bit different from traditional laboratory medicine validations in that they're statistical. The lower limit of detection that is derived is essentially an average of the lower limit of detection across the entire assay, a weighted average. And for most assays like ours, the lower limit of detection ends up being roughly 5 to 10%. If the lower limit of detection of a variant is 5 to 10%, then the detection of a presumed heterozygous variant would require about 10 to 20% nuclei. It's important to recognize here that we're not talking about the area of the tumor. You could reasonably look at the image here and say, well, that's 100% tumor, and you're basically right by area. But what we're concerned about is a number of alleles, not a tissue area. That doesn't mean anything to our assays. So what we really want is a count of nuclei. We want to exclude from the count tumor-infiltrating lymphocytes, desmoplastic reaction, vessels, fibroblasts, things like that. And we want to exclude contaminating blood and microorganisms. And so I can tell you that whenever I get a specimen submitted that's evaluated to be 100% tumor, my instinct is that that can't be true, that the person is not really doing their due diligence and appropriately assessing the tumor. Now, do we really need to know if there's 100% versus 80%? Probably not, but it's illustrative of kind of not understanding the principles at play. It is possible to enrich for tumor. You can what's called macro dissect, which would be just drawing a line on your slide around what you believe to be mostly tumor. If you do macro dissect, then you want to reevaluate the specimen for the area that was evaluated, not for the whole specimen. A specimen like this is tough, and I recognize that it's tough, and I recognize that what you have here are probably only a few tumor cells in what is mostly desmoplastic reaction, and this specimen should be ruled inadequate. I recognize that it is hard for a pathologist to tell that to a clinician. I recognize that it's hard for a clinician to tell that to a patient, especially something like this that probably felt like a substantial mass. But at the end of the day, nobody's doing anybody a service by pretending that this is an adequate specimen. And I would also point out that I have heard comments like, well, it looks like it's only 5% to me, but I'm going to send it for sequencing. And if it's below their cutoff, they'll issue a QNS. That's not true. Most assays will only issue a QNS if there was not enough DNA or if the DNA was too degraded. There is no backstop for histologic evaluation. And the problem is that you're going to issue an inappropriately negative report when what you really should have said is, we don't know, we don't have enough material to tell. So why do we do this? Why do we spend all this effort? It usually adds at least a day to turnaround time to evaluate the tissue for adequacy. That needs to be performed by a histopathologist, so that's MD time, which is on the scale of things pretty expensive. We do it, and most institutions, in fact, all that I'm aware of, do it because it's viewed as an increase in the quality of the assay that is worth the additional cost. There's a lot of concern from people outside the field about bioinformatics and inside the field too. Do I need to understand bioinformatics? I'm computer illiterate, and that scares me. So I would frame this in this way. If you are flying on an airplane, which of these three groups of people is the most important in getting you safely to your destination? Is it the air traffic control team? Is it the flight mechanic? Or is it the pilot and co-pilot? And I would hope the answer would be that they're all important. You can't just get rid of one and say it's less important than the others. If I reframe the question and say, should any of these people be required to do the other jobs? Would you expect necessarily a pilot to be able to operate as a flight mechanic, or an air traffic controller to be able to operate as a flight mechanic? I hope the answer would be no. The truth is that these groups of people need to be able to understand each other's jobs to do their jobs, but they don't need to be able to do each other's jobs. And so the analogy here to bioinformatics is that you don't need to have a huge depth of understanding of programming or of data analysis in order to interpret or use molecular data. What you need to be able to do is ask the appropriate questions of your bioinformatics team. So what even is bioinformatics? Bioinformatics is using computational tools to interpret biological data. And that word biological is critical. It's not interchangeable with informatics generally, or clinical informatics, or data analytics, or variant review, and it does not mean anything related to computers. The bioinformatics team is responsible for developing and managing the dry lab components of the assay, which are illustrated here on the right as a pipeline. And anticipating your question, yes, but what do I need to understand about bioinformatics? You don't need to understand everything about the pipeline, all the tools being used, and how they were set up. You need to understand what is the fundamental process, which we've already talked about. You're aligning fragments to the reference sequence and generating the patient sequence. You need to understand what can the assay miss, which we've already talked about, and it's because of a bioinformatic issue that the fragment is a certain length. You need to understand that the performance can vary, which we've already talked about. You need to understand that there's a background error rate, which we've already talked about. You need to understand that the VAF is at best semi-quantitative, and we'll hopefully talk about that in a second. I'm running out of time here. What is a variant of uncertain significance? Great question. Again, thank you for asking. According to the current guidelines for interpretation, which were issued a couple years ago, we use a categorization system that operates on four tiers, essentially, from benign to clinically significant. One of the important issues here is that this reporting system has dispensed with the term pathogenic in favor of clinical significance. And one of the reasons for that is things like a drug resistance variant. It's difficult to call it pathogenic, but it is very important. And so overall, it's to the good that we don't really think about pathogenicity in the context of cancer in the way we do sometimes with germline disease. It's also important to point out that the tiers, tier 1 and tier 2 in particular, are subdivided based on evidence type, not by evidence quality. So you can have really, really good, strong evidence that a variant is benign, but that doesn't make it tier 1. And at the other end of the spectrum, the FDA can have used maybe mediocre evidence to approve a therapy that still means that the variant is level A evidence in tier 1 category. So what's a VUS? If you have benign on one end, VUS in the middle, and clinically relevant at the other end, benign variants are basically those variants that are too frequent in the population to be related to the disease you're concerned with. Sometimes somebody has published something saying that, yes, this variant is benign, but it's rare. At the other end of the spectrum, something is associated with disease when there is defined literature or database evidence of a diagnostic, prognostic, or predictive association. In the middle is a VUS. It's something that we don't have enough information about to say something significant about. And there are, gradations, and so we do have these intermediate categories, but at the end of the day, a VUS is what it sounds like. We don't know what it means. Okay, real quickly, I'm going to walk through how you interpret a report. There are a lot of numbers and symbols on a report like this. What do they all mean? At the end of the day, you can ignore most of them, is the bullet point here. Any of these should be representing standard nomenclature, the gene name, the nucleotide name, and the protein variant. Notice that usually it's the protein variant that people refer to a mutation by, but occasionally, and BRCA1 is a good example, people may know the variant by its genomic name, or by its nucleotide name rather than its protein name. But for the most part, you can focus on what you need to focus on. All of this other stuff here, the genomic location and the transcript ID, are really to help resolve areas that are confusing. And I'm not going to go into how that can happen or how that can help. But for the most part, most people don't need to use this data, but it actually does need to appear in the report. And then we have quantitation. How do I use the VAF? Very quickly, the VAF is usually taken to mean approximately 1/2 of the tumor cellularity or the subclone cellularity. But this is affected by intra-assay variability that's much, much more significant in these assays than it is in most laboratory assays. Meaning, if I ran the exact same specimen twice, I could get a very different VAF value, up to 15% depending on the assay design. It's also affected by concurrent cytogenetic changes, the accuracy of cellularity, etc. And so if you're trying to answer the question, is this variant germline? 41%, it may be, but you would need to have some idea of the tumor cellularity. You'd need to have some idea of whether the other BRCA allele is lost. And you'd need to understand the variability of the assay. So the 41% could be 50%, meaning it's heterozygous germline, but it also could be 26%. You just don't know. Similarly, if you're trying to say, is a patient worsening over time, you'd need to answer the same questions. And you don't really know this either. And it's really important. I really want to convey how important it is not to overinterpret the VAF. Moving quickly here, a report should, generally speaking, also issue some kind of description about why a variant is important. This would be an example of a report describing BRAF V600E in melanoma. But you wouldn't expect the same importance in every diagnosis. These are all interpretations for BRAF V600E, but they're very different depending on what the diagnosis is. This can be a problem with industrial solutions and industrial type send-out assays where they don't have access to the medical record and can't really issue a specific interpretation, or not as specific as you can in an in-house situation. And so they issue reports that are paragraphs and paragraphs long so that they cover every possibility. And most clinicians, in my experience, find that fairly frustrating. If you still have questions, I recommend looking at the fine print of the assay. That will include a lot of information that may be helpful, like the target gene list, for example. And you can always contact the molecular professional who issued the report. At my institution, that would be me a lot of the time, but not always. And I would caution you, I'm happy to answer your questions as I'm able, but I am not the right person to answer questions about the assay that you used. Every assay is a little bit different and has its own quirks and idiosyncrasies. And you should really try to have a dialogue with the person that issued the report, or at least somebody in that lab. I'm going to skip this. So everything you ever wanted to know. I hope you understand that that was an exaggeration, but we've gone through a lot here. And I hope that this is helpful to you in whatever context you're operating in. And I'd like to say thank you to my lab, to the ASCP, to Duke, and to Dr. Jang for the invitation. And I'm happy to take questions, probably offline at this point, but I'm happy to stick around for a little while if anybody has persistent questions.
Thank you so much, Dr. Rosenbaum. We have gotten a couple of questions in the chat box as we've been going along. So are you able to stay on for a few more moments?
Yeah.
So from Jason Howard, does the reference genome that you use reflect that it is diploid, so there will be some mismatches between the parentally and maternally derived sequence?
Does the reference, okay. No, the reference sequence is a single sequence. So the problem of polymorphisms in the population is a real one. And it's worth addressing actually in the context of things that are going on in the world right now, because there are databases that exist that help you look up a variant and see if it's a polymorphism. But those databases essentially have been constructed by sequencing a lot of specimens from a lot of people presumed to be free of cancer. And there's bias in who got sequenced. And so one of the real world consequences of this is that assays that try to estimate tumor mutational burden tend to call tumor mutational burden higher, all things being equal, in persons of African or Asian descent than in persons of Caucasian descent, simply because all the polymorphisms in people of Asian or African descent have not been categorized yet. And that's not because anybody was going out of their way to exclude those populations. It has to do with history and resource availability and things like that that are well beyond the scope of the people that design and operate these assays. But the end result is, nonetheless, that people of color get slightly poorer care in that context. And so I think it's a really good example, and I'm glad that that was the first question. I think it's a really good example of how systems can be biased even without there being any particular malicious intent, or very indirect at any rate. I know that actually answers a question you didn't ask, but I'm glad I got to springboard off of what you did ask.
Okay, so I got another question of what is the cutoff of medium large indel that is easily missed by NGS?
Yeah, great question. There is no cutoff. It's going to be dependent on the read length of your assay and the tools you're using to detect the small indels, basically. And it's really hard to actually get at the answer to that question in a formal validation, because what you would want to do is introduce indels of varying lengths at the exact same position, test how reliable your assay was at picking up each of the different lengths of indels, and then do your math from there. As you might imagine, that's really difficult to do and expensive. And so most people use somewhat less rigorous cutoffs, I'll say. They recognize that there is variability in what they're able to detect, but exactly where the threshold should be usually remains a little bit mysterious.
Okay. Just a few more questions. How do you define a subclone? It seems difficult to find definition based on VAF.
Yeah, I would not do that by VAF. Like I said, the VAF is really, at best, a semi-quantitative metric. It's not to be trusted necessarily, especially if you only have two variants and you're trying to say one is at a higher VAF and one is at a lower VAF, so the lower one represents a subclone. I would say that that is grossly inaccurate. The higher VAF could be because that allele is also amplified, or it could be because the alternate allele was also deleted. It could be, if your assay is designed based on an amplicon chemistry, which we didn't talk about, it could be because of amplification bias. And so there is no way to define subclonality by VAF. Subclonality really should be an issue of interpretation. If you have enough different variants so that you have let's say 5 variants at a high VAF that's pretty similar, and 5 variants at a lower VAF that's pretty similar, maybe you can then start to draw a conclusion that the lower 5 VAF variants represent a subclone. But that's an issue of interpretation of all the data in context, also taking into the context the performance characteristics of the assay at the specific loci that you're looking at, also taking into consideration any orthogonal data, so cytogenetics or FISH results. And so it really ought to be approached only by somebody fairly well experienced and probably fairly familiar with the assay that you're talking about.
Okay, and we're getting towards a quarter to the hour. So one last question. Is the 10 to 15% tumor sample requirement also for bone marrow aspirate or peripheral blood sample?
You, uh, the, the answer to that isn't exactly straightforward. The if you're going by what I told you, that the lower limit of detection is really dependent on the error rate, then yeah, it holds true no matter what the specimen. The sequencer is not going to do better than 1 in 1,000 errors just because the tissue is fresh versus formalinized. Now, it's a little bit more complicated than that, and we didn't really talk about formalin artifact in the talk today, but formalin does tend to raise the lower limit of detection by a bit. It's usually not a huge amount if the exposure to formalin is pretty consistent. So at a high volume institution like where I work, formalin exposure is generally less than 72 hours, which is good. Um, at smaller, lower volume institutions, particularly places that may not be staffed on weekends, where specimens may sit in formalin longer, the formalin exposure becomes more of a concern and may raise your minimum input requirements.
All right. Well, thank you so much. We're about to quarter past the hour. So thank you so much to Dr. Rosenbaum. We have a lot of comments saying thank you for a wonderful talk. I'm sure if you could hear us all clapping, the sound would be thunderous. And thank you to everyone for participating.